That the pharmaceutical industry has an interest in personalized medicine should not come as a big surprise. Just last year, in 2015, fully a quarter of all drugs approved by the Food and Drug Administration had companion diagnostic information in their label and could be considered personalized medicines. This has been a growing trend for the last 10 years. And it's particularly so in cancer, where molecular diagnostics will increasingly become part of the decision-making process for use of new medicines. Overall, when you look at the industry pipeline as a whole, upwards of 50% of the combined pipelines of the pharmaceutical industry are drugs that have personalized medicine at their core and are intended to launch with a companion diagnostic. So it's reasonably likely that this trend will continue well into the future. This is a relatively new idea in that in the past, the one-size-fit-all drug was the way in which a large section of the industry operated, in part because there weren't good molecular diagnostics available to identify the patients that would either have the best clinical response or could avoid adverse events. But as the molecular biology has improved, so has the diagnostics. So now when we think about a new medicine, at the stage of concept, we start asking, who is the patient that's going to receive this drug, and how can we identify them prospectively? This works out well for both the patient, for the physician, and also for the industry. For the patients, they can be more reasonably assured that they're going to get a drug that is going to have the maximum benefit and the least side effects for them. And we've seen this not only in cancer, but in HIV drugs and anti-infectives and whole classes of medications where we can now effectively weed out patients who would have a untoward reaction to a medicine and direct them toward drugs that are more suited to them. From the prescriber's level, it adds a certain level of complexity in that you have to think about which diagnostics you're using and make sure that the diagnostic information matches the drug label. And from the manufacturer and the R&D side, this adds additional research costs in that we have to develop the diagnostics and get the diagnostics approved. But if you're recruiting only patients who are likely to respond to your new medicine, then your clinical trials run more smoothly. And so this adds some efficiency to overall clinical trial design, to patient recruitment, and to getting drugs to the market sooner, because in effect, you have larger effect sizes that are not diluted by enrolling patients who never could have responded in the first place. One of the questions that arises with diagnostic tests, and particularly in cancer, is the cost and associated with performing the test. And the cost is also borne by the patient in that if you use an invasive procedure, that's an additional burden. So this has now evolved from looking at tissue, which historically has been the basis for prescribing medicines, to now looking at DNA that can be isolated from peripheral blood. The peripheral blood is much more readily accessible, can be done quite cheaply, can be done very safely, and can be done repetitively. And this allows you to collect molecular information about a patient's tumor and use this to decide, is this the correct medicine for this individual? Now, this has been a long time in coming, but the FDA has recently approved the use of so-called liquid biopsies, collection of DNA from peripheral blood, to detect mutations in proteins that can then be associated with using a new medicine. And one good example is the use of the EGFR inhibitors. Historically, EGFR mutations were detected in patients' tumor specimens, and now there's the option to simply collect peripheral blood and test for the mutation from blood. We can reasonably expect that personalized medicine is here to stay. We're going to see increasing specificity around diagnostic criteria. Patients will be more carefully molecularly defined. And it's quite likely that in the future, the vast majority of medicines will be based on using a diagnostic. Now, this clearly has some benefits, but it does have some challenges. Ordering multiple tests can add to cost and time and can be quite inconvenient. However, there is now a trend toward multiplexing many tests onto a single platform. And we've already seen this in infectious disease and other areas, and I think this will come to cancer as well, where a single specimen can be tested repetitively for multiple genetic abnormalities or other molecular markers, and so you don't have to order each test a la carte. You order a panel, and then you figure out from the results which is the drug that's going to be most successful for this individual. So I think this will be a major sea change for use of cancer medicines, and I think it will require a new infrastructure of diagnostic testing and information management to make sure that everyone gets the tests they need, the information is available to the prescriber, and the drug is matched to the diagnostic information obtained.